In this video we will discuss about a paper that is based on chemotherapy and immunotherapy, or the combined effect of chemotherapy and the immunotherapy. So before we go into the results, let us quickly understand in brief what is chemotherapy and immunotherapy actually means. So this is the cancer cells, and in one of our videos we have discussed that how monocyte can attack the cancerous cells, or it can if it differentiate itself into the M2 macrophage, it can cause pro-tumorigenic responses, and or if it differentiates itself into the M1 macrophage, then it will cause anti-tumorigenic responses. And simultaneously these T cells will also has the capability to inhibit the cancerous cells. That is, it will not only just inhibit the cancer cells but also inhibit this cancer associated fibroblast. And how these monocytes are formed. Monocytes are basically forms from bone marrow. From their homeopathic stem cells are formed, and then it differentiate into the different progenitor cells, and these myeloid progenitor cells will differentiate into the monocyte, and then it will differentiate into dendritic cells and macrophages. And uh, what are the factors that are involved in this from these homeopathic stem cells to these dendritic cells are mentioned here. Among this, for our study we will need to know that this CSF1 also plays an important role towards differentiating from this promonocytes to monocytes, or monoblast to promonocytes to monocytes. And what is this immunotherapy or immune checkpoint inhibitor therapy? So in the normal case, what happens is that most of these T cells have these PD-1 receptors, and the cancer cells have evolved themselves in a way that they will have the PD-L1 ligand that can interact with these PD-1 receptors and deactivate the T cells. So in this case, the tumor cells will interact with the PD-1 receptors of the T cells and deactivate the T cells, and thereby helps in the immune evasion. Whereas in the case of immune checkpoint inhibitor therapy, there is a anti-PD-1 antibody is added so that it can interact with the activated T cells, and these tumor cells will not be able to interact with these PD-1 receptors on the activated T cells, and thereby whenever these activated T cells find out the unique epitopes on the tumors cells, it will trigger the immune attack. So these are the basic information. So let's first understand what this paper is talked about. The first thing that paper does is to check how this immunotherapy works on the modulates these T cells. So for this purpose they have used the mouse models, and in this case this is the experimental structures. This is the how they perform the experiment. That is, they first tumor implant, and then after day one they have introduced first they implant the tumor in the mouse, and when they see the tumor growth around 190 mm square, they consider it as day 0, and after 1 day of these observations they inject this anti-PD-L1 and then check the and then do the experiments. So first thing that they did is they check the tumor growth with this anti-PD-L1 receptors and the control, and in this case they can see that in some of the cases the tumor growth goes down, and thereafter they also check the other, since the tumor growth goes down, so they check different things, whether the CD8 plus content, they check in the tumor tissue what is the impact on the CD8 plus cells, and they can see that CD8 plus cells increased in the tumor tissue compared to the normal case. And however, when they check in the draining lymph node is, you can say, the periphery of the actual tumor cells. So in the draining lymph node, the CD18 amount is also increased. So in both the cases CD8 amount increased, and thereafter they also check the CD4 content. CD4 they also check this uh, can say the T cells uh, co-stimulator. T cells co-stimulators on the dendritic on this draining lymph node. In this case, they have seen that it has also increased in the case of anti-PDL1. So all these are accepted since the anti-PDL1 will interact with these PD1 receptors on the T cells and this prevent themselves from getting deactivated by the tumor cells. And when they check the CD8 plus when they check the CD8 plus receptors or CD8 plus signatures in the tumor cells, they can see that in the case of anti-PDL1, all these signatures such as IFN alpha, ICOS, Ki67 and ZZMP has increased compared to the control. And uh, and also in the case of this, however, they can see that uh, in the case of this CD4 plus signals will remain CD4 plus there is no change in the CD4 plus signals, and however since the CD8 amount of CD8 plus cells increased, the ratio of the CD8 plus cells by Treg ratio is also increased. That can be seen here. And when they check the CD8 plus content in the uh, overall blood, they can see that in the case when they check the CD8 plus content, they also did the experiment where they check the CD8 plus content using when they treat the mouse with anti-CD8. So in this case they can see that there are no CD8 plus either in the isotype or in the anti-PDL1, since the CD8 plus are already been removed by these anti-CD8 antibodies. And when they check the when they did this experiment with this anti-CD8 mouse models, they can see that there is no impact of the anti-PDL1 or anti-CD8 cells compared to the case where there is no anti-CD8 cells were introduced. So this indicates that it is the CD8 plus that helps in reducing the tumor volume within the mouse. And next they check the combined effect. In this case they use the chemotherapy that is carboplatin chemotherapy that has this carboplatin agent. So in this case what they did is they inhibit the they did the similar experiment. However, instead of just these anti-PD1 antibodies, they also introduced this chemotherapeutic agent that is carboplatin, and they did observe the same things, that is CD8 plus content. In this case we can see that the CD8 plus content is decreased, not removed. CD8 plus content is decreased in the draining lymph node, and this could indicate that chemotherapy has some negative impact on the CD8 plus content. And also the CD4 plus content is also decreased, whereas in the case of tumor cells they cannot see any decrease in the they cannot see any significant decrease in the uh, CD8 plus or CD4 plus content. And however and also at the draining lymph node they can see the this induced co-stimulated T cells CD8 plus is also decreased. And for the tumor cells, when they check these different signatures for the CD8 plus cells, they can see that they remains similar to the case where there is no chemotherapies, and there is no significant difference that they can observe. So since these CD8 plus cells get reduced, we since the CD8 plus cells get reduced in the case of draining lymph node, overall CD8 plus by Treg ratio is also been reduced. And when they and also okay and then when they check the tumor conditions within the different mouse model, they can see that in the case of this anti-PD-L1 and in the case of anti-PD-L1, in the case of this anti-PD-L1, they can plus carbo platin, they can see that decrease in the decrease in the volume of tumor growth, and that is an however this induced co-stimulator cells at the dendritic cell at the draining lymph node has also been decreased. So and also for this tumor analysis on the CD8 plus cells, it does not show any decrease. So this would indicates that since the decrease in the draining lymph node CD8 plus cells cause decrease in the draining immune CD8 plus cells at the draining lymph node does not have much impact on the overall immune functions when in the case of this combined therapy, that is anti-PD-L1 and carboplatin. So this indicates that the uh, tumor infiltrating lymphocytes that are present within the tumor tissues are more important, and they are the one that is responsible for this decrease in the tumor growth. Then thereafter they check whether it's the intratumoral T cells that cause this kind of tumor, this kind of immune functions. And for this, they what they did is they check the T cells. They check the T cells. They check the T cells using two different conditions. First one is the control condition, and another one is this FTY720. FTY720, what it does is it will prevent the monocyte uh, monocyte infiltration into the tumor sites. Monocyte infiltration outside the lymphoid organ. It will prevent monocyte infiltration outside the lymphoid organ. So in this case, they can see that all the carboplatin anti-PD-L1 and anti-PD-L1 carboplatins has no T cells, since the T cells are not able to infiltrate at the site of tumor. And this is after 2 days, and this is after day 9. And they also confirm this experiment using flow cytometer that the T cell receptors are absent when the cells are treated with FTY720 after 48 hours. And later on, when they did the experiment, they can see that when there is a FTY720 reagent is present, then the impact of this combined therapy has been reduced compared to the case when there is no FTY720 presence. So this further substantiate their hypothesis that it's the intratumoral T cells that are present within the T cells that are present within the tumors are causing the immune response against that leads to the decrease in the tumor volume. So that is shown here. So this FTY720 prevents these myeloid cells to come uh, uh, at the site of tumors. So next, they need to check whether this chemotherapy agents has the similar effect. If the chemotherapy has a similar effect with different chemotherapy agents, so next they check the platinum-based chemotherapy on the immune cells, and they use two different, this cisplatin and oxyplatin chemotherapy. So when they use this cisplatin, they can see the similar things, that uh, the CD8 plus T cells at the draining lymph node got decreased, both in the case of cisplatin and oxyplatin. However, there is these CD8 plus cells and CD4 plus cells are uh, all these data are for the draining lymph node. In this case, we can see that the CD8 plus cells decrease is non-significant, and also the CD4 plus cells in the case of oxyplatin decrease is non-significant. However, CD8 plus cells and CD4 plus cells are significant. And similarly, for this case, that is induced co-stimulator cells are non-significant for this cisplatin, whereas it is significant for oxyplatin. So these results are, although it looks like similar, but there is a slight changes that can be observed. And when they check the tumor analysis, uh, CD8 plus analysis in the tumors, they can see the differences. That is, for cisplatin, there is not significant difference that has been observed in the tumor infiltrating T cells, and whereas in the case of oxyplatin, they can see the significant decrease in the case of these ICOS cells. And when they check the tumor volume experiments, they can clearly see is for this anti-PD-L1 cisplatin, there is a efficiency of anti-cisplatin is low compared to the case where the chemotherapy and immunotherapies, this oxyplatin has been used in the case of chemotherapy. So, however, the results are not better like this carboplatin. However, there is some improvement in the combined therapy compared to the case of with only cisplatin. So this indicates that some combined effect of this chemotherapy and immunotherapy that might cause decrease in the tumor volume. And for this ICOS, they can see the decrease in the CD8 plus cells in the tumors, whereas for the cisplatin, they do not see any decrease in the ICOS signals in the tumor tissues. So, in order to validate that these CD8 plus cells, in order to validate the CD8 plus T cells responses anti-tumor activity in combinations with anti-PD-L1. So what they did is they use this CTX promoter, and this CTX what it does is it enhances the dendritic cells and suppress the regulatory T cells. So in other terms, it will cause pro-inflammatory responses. So in this case, we can see that for this combined therapy, there is a upregulations of the CD8 plus cells, So this is expected, and there is no change in the CD4 plus cells, since the CDX promoter only works for CD8 plus cells. And thereafter they check the tumor. Thereafter they check this tumor combined tumor. They check the CD8 plus signals in the tumors in the combined therapy, and also in the single agent therapy, and they can see that there is upregulations of this CD signals, that is IFN, ICOS and GZMB, except K67, Ki67. This indicates there is a further increase in the CD8 plus cells or tumor infiltrating lymphocytes at the tumor, or tumor infiltrating lymphocytes. And when they check the tumor volumes, they can see that there is an increase in the uh, con controlled response. There is an increase in the tumor volume, decrease in this case when the tumor infiltrating lymphocytes are high. So there is a decrease in the tumor volume. There is a more probability of decrease in the tumor volume. And thereafter they check some other reagent, that is gemcitabine, that also antagonizes the effect of anti-PD-L1 on the intratumoral C cells but enhances anti-tumor response. So in this case what they check is that when they use gemcitabine, it will decrease the CD8 plus cells. It will decrease the CD8 plus cells in the combined therapy, and also will increase the, there is no significant increase in the CD4 plus cells. And when they check for they check the tumor tissues for the CD8 plus signals, they can see that there is a decrease in the Ki67 and GZMV. So this indicates that this gemcitabine reduces the tumor infiltrating lymphocytes. And when they check this CD8 plus Treg ratio, it is also decreased, since the amount of CD8 plus cells are decreased. So in this case they expect that the combined therapy will not be able to uh, work well compared to the case of single agent therapy. However, when they check the results, they see that the combined therapy still reduce the tumor volume compared to the gemcitabine, or compared to the single agent therapy of single agent immunotherapy. So this made them to think that this gemcitabine not only reducing the CD8 plus cells, but it might also it is also reducing the some of the immunosuppressors that are present in the tumor microenvironment. So what they have predicted that the monocytes are either differentiate into these macrophages. It could be M1 phase or M2 phase. So they predicted from the literature that this gemcitabine might also working towards reducing this M2 macrophages, which acts as a pro-tumorigenic when for and helps the tumor growth. So in order to check whether this gemcitabine attacks these macrophages in this MC38 tumor bearing mice, they analyze the M2 how much M2 macrophages are present within these tumors first. So this is how they analyze this something F4 by 80, and this they check this CD206 marker, and they can see that this is the marker for M2 macrophages, and this is the marker for M1 macrophages, and this is the combined marker. And also when they check whether this macrophages has this PD-L1 receptors, and they can see that macrophages has high number of PD-L1 receptors, and also these PD-L1 MFI are high. These PD-L1 receptors, these are high. and they also have the CD45. It's also the marker for this PDL1 receptors. And thereafter what they did is they check the macrophages in the tumor tissue and also in the spleen, and they can see that in the case of combined therapy, the amount of macrophages got reduced compared to the control case and compared to the other cases. And similarly, they can see in the case of spleen also it got reduced. So from this experiment, they made a point that it is the M2 type macrophages that got reduced when the tumor cells are treated with gemcitabine. However, it also reduced the CD8 plus cells that we have seen in the previous slides. And this is the conversion of these figures. That is, most of the cells, when the person is infected with MC38 tumors, it will have this kind of compositions. That is, most of the macrophages are these M2 macrophages and the intermediate macrophages. Very few of the macrophages are this M1 macrophage. And they also further validated this result by checking these receptors, that is by checking this receptor, that is ARG1, that is also important receptors for M2 macrophage. And in this case they can check that for the CD206 plus and MC2 cells has high number of this ARG1. So this further validates their point that this MC38 tumors bearing mice has more number of M2 macrophages, and these M2 macrophages got reduced when they are treated with gemcitabine chemotherapy. And thereafter what they did is since the gemcitabine therapies also reduce the CD8 plus cells, so they need to use a reagent that does not reduce the CD8 plus cells, however it will reduce the M2 macrophage formations. So they use this anti-CSF1R receptors, and this is how the macrophages are formed. So when they target these anti-CSF1 receptors, so it will target at these 3 sites. So their main focus is to target on these sites to prevent the M2 macrophage formations. So in this case, when they check the cells, for this M2 macrophage signature, they can see that M2 macrophage CD206 plus and MSC2 minus get reduced compared to the anti-PDL1 case. However, in other case however for this CD206 minus and MSC2 plus, this does not get significantly changed, as this does not impact this, it does not get significantly changed. So it means that this CSF1, and also for this intermediate phase of macrophages, it does not get significantly changed. However, there is a decrease that can be observed. And and for the total macrophages, we can see there is a significant change, since the portion of this M2 macrophages are high compared to this M1 macrophages or intermediate macrophages. And they also validated their hypothesis that it is the M2 macrophages that is related to the tumor weight by comparing this by comparing the M2 macrophage and intermediate macrophage with this tumor weight. And they can see that with increase in the M2 and intermediate macrophage, there is an increase in the tumor weight. And finally they check whether this CSF1R has any impact on the overall tumor volumes, and they can see that this anti-CSF1R and this combined therapy has the highest impact compared to the single agent therapy, that is of anti-CSF1 or anti-PDL1. So from this they conclude their hypothesis. From this they further validated their hypothesis that it is the this M2 macrophages that are being inhibited by gemcitabine, and these M2 macrophages are working in this kind of tumors as a pro-tumorigenic factors or pro-tumorigenic functions. So this is what we have inhibited. So how this will how we will use this knowledge for the project. So in one of our videos where I have talked about this monocytes differentiating, monocyte infiltrating at the tumor sites and differentiating themselves into its M2 macrophages. This thing can be inhibited by using the Siglec-7 and Siglec-9 anti-Siglec-7 and Siglec-9 receptors. Since from that paper we have find out that this tumor cells has more Siglec-7 and Siglec-9 ligand, and this interactions of Siglec-7 and Siglec-9 with the tumor cells helps them to differentiating towards M2 macrophages, M2 phenotype macrophages. However, if these receptors are blocked, then they will not be differentiated into the M2 macrophages, whereas they will differentiate themselves into the M1 macrophages, and that is beneficial as it will have anti-tumorigenic effect. So we can use these information that we have discussed right now in the following scenario. That is, we can take the human blood cells and isolate monocytes and T cells, and they use the knockout libraries that we have generated. Two different knockout libraries, that is, one is essential gene libraries that are important for pancreatic cancer, and another one is essential gene libraries that are important for the cell survival. So we can use both the O-glycan libraries and essential gene libraries separately in this experiment, and use these receptors, that is anti-Siglec-7 and anti-Siglec-9 or CSF1, and along with this anti-PDL1, and after some time we can check which are the cells that are survived and find out the guide RNA to help us to know which knockout genes helps in this cell survival, so that we can finally make a point that these knockouts are more prominent, these knockouts are more important, these knockouts are more important for this tumor formations. And if someone has these kind of knockouts if some person has these kind of knockouts, then it is difficult for them to get a cure from this approach, that is using combined immunotherapy and chemotherapies. So that's all from my end. I hope you got an idea about this project or this paper. If you need to understand more about this paper or this experiments that we have results that we have discussed in this video, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.